Hey everybody, welcome to lesson 9. Today we're going to be going over microbial growth and what impacts microbial growth has in medicine and science and why it's so important. So the first thing we really have to understand with microbial growth is that there are a few different ways that bacteria will replicate, but the main way is going to be binary fission. This is where one bacterial cell then grows into two bacterial cells, and they're copies of each other. So it's going to be the same DNA and essentially the same overall organism, just split into two. So this is what we mean by, getting individual colonies. When you get an individual colony of a bacteria, if it's undergoing binary fission, when it replicates, you're able to say all those have the same DNA. This is really, really important for drug development and drug manufacturing because if you're manufacturing a drug in a bioreactor with bacteria, it all has to be the exact same DNA and everything else. So binary fission is great. It starts with DNA replication. It replicates inside the single unit. The cell elongates and then it'll form two daughter cells. Now, you might be thinking, this sounds a little bit similar to things that we've talked about in, general human biology, because this is just generally going to be the way that all cells replicate, is they're all going to split and split and split and split. And that is the same in a lot of mammalian cells. Mammalian cells get more complicated because of regulators over bacteria, but that is how most cells are going to replicate. Now, different bacteria will have different times it takes for this doubling to happen, and that's called your generation time because every time you double, that's a new generation. So you go from one to two, from two to four, that's two generations, the parent generation, the first split and the second split. So two generations. This is important for a lot of different reasons. One being that if you're using any kind of bacteria in a bioreactor for manufacturing, you want very fast generation time. And this is why E. coli is used a lot for doing replication. Even in food processing or bioprocessing, you're going to use something like that. Or maybe yeast. Yeast also has a fast generation time. Yeast is a eukaryote, but still replicates very quickly. It's also going to be very important for antibiotic use. If you have a fast generation time, you know that you need antibiotics really, really soon. You don't want to wait and see if someone stops getting sick. It's going to spread easier. There's all of these things that go into it. And then it's also going to impact the incubation time of a disease. Because if you have something like an E. coli infection, that's going to come on really, really fast from person to person. But mycobacterium, which has about a 15-hour generation time, that's going to come on over the course of weeks after your exposure because you've been exposed. It's growing, but it's generating so slowly, and then it forms this critical mass where you get sick. And that takes a long time versus E. coli. And an E. coli infection, it's turning over every around 20 to 30 minutes. So you can see how crazy of a difference that is, 20 to 30 minutes versus 15, 16 hours. And then the other thing is when we're looking at ease of studying microorganisms, ease of studying a lot of different things, we want these fast generation times because we can do experiments where we look at, okay, we have N generation and we want to get 1,000 more generations. Well, with something like Mycobacterium tuberculosis, that's years. But with E. coli, because it's 20 to 30 minutes, that is maybe, a couple months, even in unoptimal conditions. So it's a very big deal. Now, when this generation is happening in a mass scale inside a contained uh, system. So something like a flask or a test tube, anything like that, you're going to have a very specific growth curve that also maps to different microorganisms. This starts with a lag phase. This is where they are adapting to a new whether it is substrate that they're being grown in or a new environment, if it's happening out in the big wide world, there's this lag phase. So they don't really grow. They're getting used to where they are. They're making sure that they can actually replicate and have all of the energy they need to replicate. And that kind of goes back to what we were talking about in the previous lessons of getting all this energy from different sources. They want a source of energy. So they're growing, growing. maybe very, very small or not at all, and then all of a sudden they just shoot up because they start dividing as fast as they can. They're very metabolically active, and at this time, they are antibiotic sensitive to what we call bacteriostatic antibiotics. So there's two forms of antibiotics that are typically used. One is bactericidal, cidal kill, so it kills the bacteria. And then there's bacteriostatic, it just stops growth. So your body can take care of the bacteria on its own. So during that exponential growth phase, those bacteriostatic antibiotics are fantastic. And then it reaches this stationary phase where you get a division rate equaling the death rate. So it's not growing, grows really fast, stops growing again because the amount of replication is equal to the amount of death. And this starts occurring because they're running out of resources. So earlier I mentioned a closed system. That closed system doesn't have to be completely closed, it's resource dependent. And then after it starts dying off, you get this, this hump that it gets over, and then they all start to die. And this is called the death phase, obviously, because they're all dying. And the population, like it exponentially grew, it's exponentially dying because they're out of resources. And this growth curve is directly linked to your infection progression if you're infected with a microorganism. The lag is what we call the incubation time. So that flat phase is when it's not really growing much or not at all. And then you have this log phase, which is your illness. You have a stationary phase up at the top, and that is your persistent infection. I'm sure, like me, you've had a time where you've been infected and it's 3 weeks and you just have this little bug and you're not getting rid of it. Well, you are getting rid of it, but you're in that stationary phase. And then the death phase happens, and that's your recovery time. That's when you're feeling a lot better, but you're still a little sniffly, or you're feeling like, I'm doing pretty good, but if you push yourself too hard, you start feeling sick again. And that's during that death phase. So it maps one to one with what we feel as people when we get sick. And this also goes into how we can kind of count bacteria. When we're counting bacteria indirectly, we usually talk about turbidity. So this is the cloudiness of a solution. And that can be tested with a spectrophotometer, and it is a quick approximation of how much bacteria is in a culture. It doesn't really tell you, though, if it's growing, stationary, or dying. So you don't know if it's in that log phase or if it's in that death phase. You just know there's a lot of bacteria there. So it is an indirect method. You can't be 100% sure what's going on. When we look at direct methods, there are a couple different ways. You can count with a microscope. That's kind of a pain. You can do serial dilutions and then grow. This is called plate counting. So you can get a certain number on a plate, usually between 30 and 300 that you can count. You can look at the dilution factor, and based on that, you can calculate how many bacteria are in that actual growth media that you're using. You can do a couple other things, but those are typically going to be the two that are used the most. Honestly, plate counting is used, in my experience, the most just because it's easy to do. It's pretty direct. Um, you just make, 5 or 6 plates. You say one of these will grow in the range I need to. You throw them in an incubator. You come back tomorrow. You count, and then you know exactly how much bacteria you had. And this goes into what we were talking about in the last few weeks about things like plaque assays and viral assays. This is similar because you're just counting how many colonies there are, just like you're counting how many plaques there are for viruses. So those overlap and are essentially, at the end of the day, the same method. Now, counting, pretty straightforward, but not really important outside of doing some sort of analysis in a hospital lab or doing, direct science in an academic lab. But there are a lot of other things that happen during the growth that we need to discuss. One of them is the formation of biofilms. So in the lab, we don't really get biofilms because we don't let microbes get out of hand. It's always being used for a purpose. And we're not really dealing with biofilms because there's a lot of turnover of any microorganisms that are being used for research and science. But these biofilms are a microbial community that is encased in what we call extracellular polymeric substances, EPS. So you have a bunch of bacteria, they're growing, growing, growing. they are grown into a space and then they start making these substrates that make it easier to live in that space. So they attach to something, like the the underside of I don't know, the corner of your sink, let's say. And then that attachment kind of becomes reversible, irreversible because of these extracellular polymeric substances. They're essentially glue. So they glue themselves to a space and then they start growing and dividing and they keep up that production. And then at a certain point, they've totally taken over. So that's how those biofilms are made. And if you've ever been cleaning something that was like back in a corner and you're like, why is this so hard to clean? It's because there's essentially a bunch of microbial glue, this EPS, all over where you're trying to clean. So you've got to put in some extra elbow grease, right? Then there's some advantages and disadvantages of this. The advantages is adherence. You can share nutrients between all of the different bacteria in this biofilm cluster, and you have a lot of resistance to disinfectants and immune defenses. Obviously, bacteria aren't made to fight disinfectants. It just works out that way. What this biofilm's made to fight against is your immune defenses, the immune cells penetrating in, getting in there, and destroying everything. This is a major problem in the medical setting because when biofilms occur, they're hard to clean, and there's usually going to be leftover bacteria in general. So, on things like catheters, prosthetics, stents, anything that is going into a body, if it has a biofilm, then you can induce an infection just by exposing the person to that. And that is very, very dangerous. Then, on top of this, there are different requirements depending on where these microbes are living, how they're living, and what they're taking in. So, everything needs nutrients, and we've talked a lot about how different microbes will use different nutrients. Sugar's the best. Everyone wants sugar, but there's fungus in Chernobyl that is growing using the radiation from Chernobyl. There's extremophiles on superheated vents in the middle of the ocean. So there's all of these options for taking things in, but we need a way to categorize. Science is about categorizing things, putting them into buckets, and knowing when those buckets are overflowing and overlapping, right? So we're trying to put things into categorizations that make the most sense for us and our daily lives. So one of the ways we look at that as oxygen breathing organisms is, do microbes need oxygen? There's obligate aerobes. Obligate aerobes need oxygen all the time. So if you take away their oxygen, they die. We're obligate aerobes. We need oxygen at all times, or obviously we're going to die. Um, there's microaerophiles, which need low oxygen. So they can survive when there's not a lot of oxygen, but they still need some oxygen. Then there is facultative anaerobes that can grow with or without oxygen, but they prefer oxygen. This is something like E. coli. And why E. coli can be great for science and medicine, but awful for infection is it is this facultative anaerobe where it's going to be able to survive in a lot of conditions. There's aerotolerant anaerobes. These ignore oxygen completely and use fermentation. So, strains of Streptococcus are going to be these aerotolerant anaerobes. They're going to be able to survive in oxygen, but they're still going to need to use fermentation to actually grow. And then you have obligate anaerobes. These are actually killed by oxygen. A great example of this is Clostridium perfringens. This is a infection that affects chickens. I actually made a vaccine against this as my master's thesis way back when. And it's really, really bad because it affects the chickens. It kills them. It makes them lose a lot of weight because it's in their gut where there's no oxygen. There's just this milieu of food. So they replicate really fast and they make the chickens really, really, really sick because of it. But if you tried to take a sample out and keep it alive and it gets exposed to oxygen, it's dead. So there's a lot of interesting things that really impact where microbes are the most successful and why we get these microbes that are pushed out more and more and more into these extremophile situations. Because there's always a niche to fill, and they will always fill that niche. Um, Another example of this is pH. So you have neutrophiles, which want to be around pH of 7. E. coli wants to be around a pH of 7. It's a neutrophile. It wants to be at that pH, and that's why it can actually infect our blood because our blood pH is around 7.4 to 7.6. So you can get an E. coli infection in your blood. Really, really bad if that happens. There's acidophiles. These are bacteria that thrive at low pHs. So we're talking about 3 pH or under. That's really, really acidic. And these are going to be a lot of things that live in the stomachs of organisms. If you thought that you don't have a microbiome in your stomach because all the acid would kill everything, got bad news. There's plenty of microbes that live in an acidic environment, and it's great that they're there. It's great you have a microbiome, or else it would be dysregulated and you'd have a lot of problems. Similarly, there's alkophiles, alkalophiles that are going to thrive when it's above that pH of around 9. So the pH scale goes 1 to 12. Seven's in the middle. That's what we want usually as people, and a lot of microbes live there, but there's, again, those extreme files get pushed to the edges. The same thing happens with temperature. Same thing happens with pressure and light. So some microbes thrive under extreme pressure. They're called usually barophiles, barometric pressure being the source of that. And they're the ones that are around these deep sea vents. They're just under so much pressure and they gotta live there. And there's a lot of more complex life that lives at those depths as well. And similarly, they're barophiles. They have to be at that depth to operate. Phototrophs, they require light for photosynthesis. That's pretty straightforward. We talked about algae a lot. Algae is an example of that. Um, and then there's, of course, temperature. You have whether it wants to be at very low temperatures. Optimal thermophiles are about 50 to 80 C, and these are things that are going to want it very, very hot. And they, luckily, because they want it hot, can't infect us because we don't run that hot, and that's their optimal uh, functioning level. And then there's hypothermophiles. This is above 80 degrees Celsius. So this is boiling almost. This is simmering hot water on the stove is where they love to be. And where we get Taq polymerase, which is used, millions of times a day in PCR reactions, is actually from Thermus aquaticus, and that is a hypothermophile. And that's why that Taq polymerase is wonderful because we can use it and do polymerase chain reactions at a high temperature, which makes them happen faster. It would be a very slow process. It actually used to be done by hand and take multiple days to run a PCR. Now you can run a PCR in an hour in a machine. It's kind of wild how much it's progressed. Um, We've talked a lot about media as well, and we're going to get into different kinds of media in this lesson. But it's kind of just one of those lists that you have to memorize. Really, the media you use is going to be different from every other microbe to the next. There's some that microbes just tend to like to grow in. So super optimal condition broth or SOC media, SOC, that is a media that's used widely because it's just this highly enriched media that a lot of microbes like to grow in. Another example of a enriched media is blood agar. So it just uses blood from usually young cows, and it's got everything in it that things that live in blood or want to eat blood are going to need. So that's used a lot as well. But like I said, there's a lot of different culture medias that are used. It's going to be very different from microbe to microbe, but there are some usually those all-purpose or enriched medias that are going to be used very widely in science and medicine because they can grow so much on them. You wouldn't want to be running patient samples, for instance, in a media that's only going to grow half of what they might be infected with, right? So you're always going to use an all-purpose growth media, and that's going to be usually like nutrient, agar plus broth is it. Very simple. Or something like an enriched media, it's got that blood in it, so it's going to grow anything that might be a blood infection, for instance. So there's a lot of ways to deal with that. For this lesson, I think it's a lot less memorization from last two lessons. I know all the metabolic stuff is a lot to take in, so I hope this is a little bit of a reprieve. But you're mainly going to want to be able to explain binary fission, that growth curve there will be an image of, uh, and then also explain how all these different interactions of microbes and their environment impact health and science. So just keep that in mind while you're reading through everything. And hopefully, this is, like I said, a little bit easier to wrap your head around than last lesson. I know we've kind of visited all these topics before in one way or another. So hopefully, it's a quick, easy click. But if you have any questions, reach out, let me know. And I hope you have a fantastic rest of your week.